Project 4 will be a little bit different than the other projects, although we will use Mega a little bit. It won't be primarily based in Mega. Now, along with that will probably come some technical glitches or problems with the different files that you'll need to use. So if you have a problem, if you get stuck, send me an email, log on during a office hour. I'm happy to help you and walk you through it. That's just kind of part of it. It's one of the downsides of this project. I've tried to keep them pretty simple as far as the amount of software that you need to use. And although Mega has its problems and glitches, it can do many things just on its own. We can't quite do it for this. So just be aware of that. Contact me if you have problems. In the Project 4 files, you'll have a link to a OneDrive folder, and you should have full access to it. What you want to do is once you access that folder, you'll want to download the files that you will need for this project. Okay, so each of you will be assigned a transcriptome. There are 8 different transcriptomes we're going to be working with, and I've got this transcriptome 0 that I'm going to use as an example. So you'll need to download the transcriptome file. It's a rather large file, so it might take just a little bit of time. And then these other files, which are smaller, you'll need to download them also. So open up the, let me find the one here on the bottom. Open up the transcriptome assignments folder. So download that one first. Figure out which transcriptome you've been assigned. So for instance, Julio would be transcriptome number 2, and that would be the one that he would be working with. So once you've identified the transcript that you're going to be working with, you need to find that one. I'm going to do 0 because it's an example. Find that one, and then download these other 5 files. So everything from Make Blast database. This one is an accessory one. You don't absolutely need it, but it will be helpful. You need to install it on your computer. If you don't have admin access, if you're on a university computer, you will be able to install it, but you have to do the run elevated. You don't absolutely need it. You can use any text editor. And then these other 3 files. Now here is a note, and I'll post this in the instructions for this worksheet also. You will need to be on a PC. So these 2 applications that we're going to be using, and Note Tab Lite also, but that's an optional one. But these 2, TBlastX and Make Blast DB, those are on a PC format. So you'll have to do this on a PC. If you're a Mac person, you'll need to borrow a PC or find some time to go to a university computer lab. Okay, so download those 5 files here and then whichever transcriptome you're going to be working with. Okay, so here is my folder that I'm working with. Again, put them anywhere. It's just convenient to put them all in the same folder. I would suggest that you double click on this and install Note Tab Lite. I'll show you. I've already got it installed, so I'm going to delete that. But if you install that, that'll make it a little easier. But you can use any text editor. Just Note Tab Lite makes it a little easier for the work we're going to be doing. Okay, so the very first thing we need to do is I'm going to take a look at this file. This is an alignment, and what I did was I downloaded 3 genes. These are receptor genes. They're not really closely related to each other. If you look at the protein translation and try to do an alignment of these genes, you'll notice that there's really not much similarity at all. So other than being receptors, do that again real quick, sorry. Okay. So here we go. Other than being receptors, so generally they may have a small domain. I don't even see much conservation at all. One's longer than the other 2. There's not really any. So these are very, very distantly related, maybe even no connection that we can find related genes. And again, they may have some very small domains or motifs in common, but not much. So 3 very different receptor genes. And these are all from grasshoppers, Locusta migratoria, Schistocerca gregaria. Those are 2 synonyms. So these are the same. All 3 of these are from the same species, a locust or a grasshopper. Okay. Now what we're going to do is we are going to look for homologous copies of these genes in these transcriptomes that I have. And those transcriptomes that you have been assigned are each from a different species of insect. And they're going to kind of remain mystery insects. That will be part of the assignment at the end for you to maybe take a guess, estimate what insect those are from. So we'll walk you through that. So these are what we're going to be working with. Now I've saved these in just a simple text format in this receptors.fast file. So if you open up that file, and I'm going to use Notetab Lite, but again, you can use any text editor, it's just a simple text file. What you will find is simply the name of the gene, followed by the gene sequence. This is just the coding sequence, and we've got that for all 3 of these genes. And notice one's longer than the other 2, but that's going to be our query file, or the file that we use to search for our homologs in our transcriptome. Now, to be able to do this, we need to turn your transcriptome into a searchable database. There need to be some support files to do that, but we can do it all locally on your computer. So instead of sending these files off and saying, tell me everything that's been published historically that's close and similar, all these DNA sequences, I want you to say, I have a transcriptome from a specific organism, and I want you to find me close homologous matches to these genes in that transcriptome. Okay, so that's what we're going to be doing. So here it's going to get a little tricky. If you've never used command line, I'll try and be as straightforward as possible. You should just be able to follow along really, really easily. If you get stuck at any point, reverse the video, watch it again. If you're really stuck, send me an email or log on during office hours and we can walk you through it. We can share screen and help you get through it. Okay, so to do this, I need to open a command file. So a command prompt, I'm going to do that. And then I need to navigate to this folder where all of my files are. To do that, we're going to type CD, that's for change directory, and then put a space, and then all I need is the address. The easiest way to do that is I'm going to right-click on and then copy the address for the folder that I'm working in. And then I'm going to go back to this command prompt, and I'm going to paste it in. You can do right-click, or you can do Control-V. Either one will paste in. And then you hit Enter. And so now I have changed, so I am located in the directory that I'm working with, okay, whatever your directory is. Now that we're here, we can enter commands to do different things with the items in this directory. Now, the first thing that I want to do is I want to make a BLAST database. Now, the software to do that, the little application to do that, is already in the folder. You should have downloaded it and put it in this folder. So if you don't have this make BLAST DB executable in the folder, you're going to be in trouble in it. You'll get an error message. So, but I've got it there. Everything should work. So I'm going to put make BLAST DB. That basically says open the program that's titled make BLAST DB. And then what I want to do is, let me get the commands here. I'm going to get the commands here to remind me what commands I need to use. I think they are, sorry, they're right here. Let me just open this one really quick. Okay, so to enter a command in this program, you put a dash, which says that it's going to be a command. The first command we're going to do is DB, which stands for database. We want to make a database out of, and then you just put in transcriptome 0.fas. So basically I said we want to make a database out of this transcriptome. Let me get back to where we were. Out of this transcriptome 0.fasta. And so we need to make sure, just double check, make sure you've typed it in exactly. If you make an error, it will say, you know, if you type in a wrong letter or just make a small spelling error, it will say, hey, I can't find that file. So here I've done make BLAST database. The database that I want to make is going to be from transcriptome 0.fasta. And then I want to make sure I tell it the type of database. So DB type is the command, and NUCL stands for nucleotide. So those are all the commands that we need to make a searchable local BLAST database. We're going to hit enter. If everything goes well, oh, look, I've got a little error there. Let me see what I did wrong. Oh, sorry. It was the wrong command. So let me go back. It's not DB, it's in. So I just put the wrong command, so I was looking for that command. So the command is in, meaning this is the input file to make the database out of, and it's going to be a nucleotide database. Okay. So with that, you'll see it will to it. If everything works well, it will tell you that it's added all the sequences, and then you'll get the command prompt back. That means everything's been executed successfully. And now in your folder, you should have 3 additional files. These are the support files that will make this transcriptome 0 a searchable database. Okay, so you should see those 3, confirm that those are there before you move on. Okay, so the next thing that we need to do now that we've made it a searchable database is we need to do a search using this file. This is our file here, receptors.fas, that has those 3 DNA sequences. We're going to use that as a query to search our newly made local BLAST database. So to do that, we're going to use this other program that I've put in that folder. You should have downloaded this one also. So if it's not there, you'll have a problem, but we've got the folder tblastx executable. So we're going to say tblastx, use that program, and then we're going to give it some commands. So this is where the db command comes in. So the database that we want to use is that same one. So we're going to say database transcriptome 0.fasta. That's the database that we want to search. And again, just double check, make sure you don't put spelling errors. You'll get a message, an error message if you have one. Then we want to give it a file that we can search. And since I have 3 different sequences in your query file, it's going to do a BLAST search for all 3 of those sequences. So query is the command, and then we just want to give it the name of that file. And I've called it receptors.fas. Okay, so again, we'll double check, make sure, okay, there's my query file. So I'm going to use that query file to search my database. And then the last command that we want to give it is an out command that will tell us where to put it. Sorry, OUT. So -OUT, and you can give it any name you want. I'm just going to give it a nice easy one. I'm going to call it receptors dot results dot out. Again, anything you want as long as you can keep track, that's fine. Okay, so we'll hit enter, see if I did everything correctly. And if you did, it's going to work on it. This one, depending on your database size, might take anywhere from a few seconds to several seconds, probably less than a minute. And what you will have now is an out file with your results written into it. Okay, so we made a local BLAST database. We took some grasshopper genes that I wanted to see if there were homologs of in my local transcriptome. And now we're going to look at the results and see what we found, whether or not we have results. Okay, now this is where you're going to want your text editor. So I'm going to use the Note tab one. If you haven't installed it or don't want to, that's fine. You can just use Notepad on the PC. I think you can even open them up in Word, although it's just a simple text format, so it might be a little, you might have some problems opening it up. So the text editor is probably easiest. So I'm going to drag this receptors.results.out into my text editing program. And now I have the first query, which was Locusta migratoria, insulin receptor 1. And then I've got a list of results. Now, we want to look primarily at this E value. The lower that E value is, the more likely it is that we have a homolog. If it's a 0, that's really, really good. It's almost certainly 100% chance that we found a homolog. And notice that we have a couple with really high scores here. And then the scores start to get a little worse. This, by the way, is just an annotation for 3 times 10 to the minus 60th. So really, really tiny number. That's a really good score. So very likely this is an ortholog, an exact descendant of that same gene we're looking for. Now these others may be in the same gene family. They may be paralogs. We might want to evaluate and do a little bit more in depth if we're really, really doing a long analysis of this transcriptome. But what I want to do for each of you, I'm just going to have you take your very top score. Okay. Now, if it's below minus 50, there's a very good chance it's not the gene we're looking for, but maybe a member of the same gene family, or it could be the same gene, but really, really highly divergent. But I think for most of these, I haven't actually looked. That's going to be part of this assignment, is me evaluating what you've done and comparing how you did with other students. But most of these, I think you will find it. But if you get a very high E value here, something higher than 10 to the minus 50th, there's a chance that it's not the ortholog, maybe a member of the same gene family. Okay, so what we are going to do now is we would like to find this transcript. Now the names are given semi-randomly. There's a systematic way to do it, but there's not anything meaningful. But I want to find transcript number C9609G1I1 in my original transcriptome. So to do that, I'm going to open up my original transcriptome also in this text editor. And these files are a little large. It's a big data set, so it might take a little while to open it up. So there, that wasn't too bad. So I want to look for this match. So what I'm going to do is I'm going to highlight the name, and you can do just the first part of the name. You could do more of it if you want, but that part enough will be a unique identifier. And yours might have a different prefix. The numbers are going to be different, but just go ahead and highlight that name. I'm going to copy it. You can use Control-C, or if you like better, you can right-click copy. Control-C is a nice little shortcut. So Control-C, and then what I want to do is come over to my transcriptome file, and I want to search for that transcript. So once you get here, press Control-F. That brings up the find, and then you can either right-click paste or just Control-V, paste the name in there, and then let's find our next match to that. So there it is. So I have the gene name that's been assigned to it, and then the potential transcript sequence. Okay. So once I've found that, I want to highlight both the gene name and the entire sequence, and I'm going to copy. So again, right-click, copy, or Control-C. I then want to make a brand new text file. One of the nice things about NoteTab Lite is you can have multiple tabs open, so we can switch back and forth easily. And I want to paste in that gene. Now, because it's just been assigned a random gene, I'd also like to make sure that I know which one this was a match for. So I'm going to come back here, and then let's look at which of these it was a match for. So in my results file, I've got, Insulin Receptor 1. So just so I can keep track of which one this is potentially a match for, I'm going to put Insulin Receptor. So there we are. I have located a really good match, most likely a homolog to this insulin receptor. Let's look at the others. So I'm going to come back to my results. And it's a big file, so I could scan down until I found the next query, but probably the easiest thing for me to do is to say, well, let's just find where this next query is. So here's the next one. This is an aryl hydrocarbon receptor. And notice the match is not quite as good, but it's still a pretty high number. Now, the other thing is this is a shorter sequence that it retrieves, so it's very likely I don't have the complete gene. I might only have a partial gene for this one. If it's longer than 1,000, there's a good chance I have the whole coding region. But this one I might only have a part, but it's still a very good match, so I think we're okay. We don't always have the complete gene in our results. So same thing. I'm going to highlight the name of that gene. I'm going to come to the complete transcriptome. I'm going to paste in the name of that gene. Let's tell it to search the whole document and find where that gene is. Here it is, a little bit of a shorter piece. I'm going to copy that out, and then I'm going to come back here to my other file. And you can put extra spaces if you want, or you can just put them one right after the other. I'm going to put the next one, and this is my aryl hydrocarbon receptor. Okay, so now I found a match for 2 of my 3 genes. We're going to repeat the process. I'm going to come back here, Control-F, see where the next query is. There it is. This is a glutamate receptor. And again, this one may be, it's a pretty long piece, but for this transcriptome at least, I don't have a really great score. It is above -50, but not a whole lot. So anyway, yours may be as good, might be better, might be worse. So whatever is there, that's what we have, that's what you're looking for. So whatever the highest match is. So I'm again going to now search for that one. I'm going to do it in this one. Search the entire document. Here is my next gene. Highlight both the gene name and the gene sequence. Copy. And then paste it. And then again, just to remind me, this is a glutamate receptor. So now what I have is a text document with a name, a little bit of a description, for each of these 3 best matches for these receptors in this mystery transcriptome, this insect transcriptome that I don't know what it is. So before we lose that, let's go ahead and save that. It's a shortcut here. And I'm going to put it in the same folder here. I've got it in here. Okay, so we'll just give it a name. We'll call it receptor matches. You can call it whatever you want, as long as you know where it is, and we'll save it there also. Or how about this? Receptors confirmed. Now, we're not totally confirmed yet. I'm going to show you the next step of how we will confirm that. I would recommend, rather than just giving it the default .text, let's just give it an FAS ending. That's basically for FASTA format. That way it'll simplify things and allow us to open it easily in Mega software. Okay, so if you don't have Mega, you don't absolutely need it. I know some of you might be using a different computer and you might not have Mega available on that. That's okay. We can do everything without Mega, but Mega will make it a little bit easier. Okay, so now what I want to do is confirm and maybe get an idea about what other matches might be out there on the full database that may match these. So I'd like to take each of these and search these on a general BLAST database just to see what close matches. And that might also give me some clues as to what organism this is. Okay, so let's take a look. We'll do insulin receptor number 1. Two ways to do this. If you've got Mega on your program or installed on your computer, you can just open it up. And so if you click on the file we just made, which is receptorconfirm.fas, it should open that up right as an alignment in Mega. Here we are. If you don't have it, that's okay also. Now, because these are transcripts, there's probably some untranscribed region on the beginning, or if it's an incomplete one, we might have just a part of the gene, and it could be in any of the 6 possible reading frames that we have the correct amino acid sequence. So we don't have these in correct reading frame. All we have is the raw DNA sequence. So because of that, we're going to do a little bit of a different, or we're going to be careful about which BLAST search we use. So I'm going to highlight this first one, and now I'm going to go to the BLAST search option in Mega. And if you don't have this, you can just open up a web browser. Open up any web browser and go to BLAST. And you can just paste that sequence into the BLAST window once you get there. So you're welcome to do that if that works better for you. Okay, I had a bit of a little issue with my internet connection. Let's see, hopefully it's resolved now. So we're going to take this first match to our insulin receptor. We're going to highlight it and do a BLAST search. If you don't have Mega, again, you can cut and paste that into a BLAST search just through any old web browser. The key thing that we want to do is we want to use the BLASTX. Now, BLASTX is going to search all the protein databases by translating this nucleotide. So it's going to find the largest open reading frame. I think it actually does all possible reading frames and looks for significant matches. But this way, we have a better chance of finding all of the close matches because the protein sequence is going to be more conserved than the DNA sequence. Okay. And for this one, if you'd like to speed things up a little bit, you can actually modify it. Because we know this is an insect sequence, you can search just among the insects, so we're not going to search the entire database. We'll only look for sequences that come from insects. So we'll do a BLAST search here. This should take under 30 seconds, depending on how busy they are. It might take a little longer, but so let's see if this gives us any clues as to what this gene is. Now, we would expect it to come back with other insulin receptor genes. If we find an exact match, we might even know what organism it came from. But certainly if we start to get a range of organisms, if it's a wide range, then maybe we're not very certain about what organism comes from. But if we get a narrow range where we might have just, maybe all from the same family or the same order of insects, then we have fairly high confidence that we know what organism this insulin receptor comes from. Now, whether or not we get a exact match is going to depend on a couple of things. It's going to depend on how much work has been done on this particular gene. Has it just been worked on for the model organisms and sequenced and understood there, or has it been maybe of more interest and worked on more widely? It's also going to depend a little bit on how quickly this gene is evolving. Is it evolving at a very, very fast rate, or is it evolving more slowly? And if it's evolving slowly, then we should get much more close matches. Okay, so here are our results. We have a couple of different ways to look at those results, but notice we have a very, very high score from Timema. Now, Timema, if you look at it, click on it, that's a stick insect. Platella is a cockroach. Here is that same sequence we started with. So although we found a close match to the sequence we started with, it's not the closest match overall on the database. But these are all very, very high scores. So we can be very, very certain that not only have we indeed found this insulin receptor, insulin-like receptor means that they've verified it just by sequence similarity. Now notice some of them say unnamed protein. These are from large-scale products where they just kind of did a whole bunch of genes from maybe a single or a group of transcriptomes. They put them online, but they never did much work to completely identify them. But notice everyone that is identified is an insulin or insulin-like receptor. And again, the like means we're pretty certain it's that sequence, but we haven't verified it other than just based on its sequence. And so we have stick insects, cockroaches. As we get down, we have some termites down here. If you don't know these, that's fine. So fairly certain that it must come from this group of organisms that are listed here. So if you're not familiar with insects, you may not be really super familiar, but we could say, oh, it's one of these polyneopterous insects. And I don't expect you to know insects for this group, but just to give you a little bit of an insight, the polyneopterous insects include roaches, walking sticks, praying mantises. What else do we have here? We have the crickets and cockroaches. So we're fairly certain, because these are all of our very close matches, that it's a member of that group. Although we don't have maybe enough to say, oh, look, it's most closely related. All the really close matches are from walking sticks. So maybe we could say it's potentially a walking stick, or maybe the closest relative of a walking stick, which is an Ambioptera. Web spinners, you're maybe not familiar with them. But again, not really important, but you can maybe say a little bit about it based on that. Okay, so let's do it, and I'll go through these rather quickly, but let's do a similar thing for the other two. So we have a aryl hydrocarbon receptor. I'm going to blast this. And again, it should default. Oh, it doesn't. We'll speed it up just a bit by confining our search to just insects. And while that's going, just to speed things up again, I'm going to get another window open here where I'm looking for the glutamate receptor. So when I do this, because I've got two separate runs going at the same time, oops, the other thing is I forgot I'm doing a blast N, which may very well work. We'll go ahead and let it run. I forgot to change it to blast X, but blast N's a little faster. So let's see what we've got. Cryptotermes, so again, termites, fairly high scores. Again, above 50 is a good sign. And for some of them, let's see, we can't see what gene name that is there. Let's see if we can click on it. Yeah, uncharacterized. So we know it's a protein. It was deposited online as a protein, but was never really described. But the ones that do have descriptions, there's our one we used to search for, aryl hydrogenase or aryl hydrocarbon receptor, aryl hydrocarbon. All of these that have identified are aryl hydrocarbons. And again, our closest matches are just right where we would expect them to be among those termites, the cockroach or the grasshopper. And then we start to get a little bit farther outside of that. So that one looks good. Let's try this last one. There we go. This one I will change it to the BlastX search. Looks like it was not really that necessary, at least for that second gene, the BlastN, which is just searching the nucleotide database without doing any translation, and just looking at nucleotides worked very well also. Okay, so we'll let this run, and then I will walk you through the final parts of what you need to do to complete this assignment. Okay, so here's the blast search result on our final identified transcript. And same thing, time EMA, right? So it's a really good indication that's probably very closely related. Notice these E values are very, very high for these. So these glutamate receptors, time EMA, there's a termite. I don't know what that one is. We could look. I don't recognize it. Oh, so that's a hopper of some sort, like a true bug. Oh, sorry. Okay, let me close that. Okay, so anyway, good confirmation. We've found matches for these genes. Again, you may or may not find, there's a really good chance based on the samples, because they're both similar. They're all the same tissue, but from different groups of insects. So I will go through here really, really quickly. There is going to be a short report. You're going to want to watch the rest of the video to help you walk you through, make sure you get everything complete. Okay, so once you've completed that, once you've taken your best hits for each of those queries and then search them on the BLAST database to see if you can find anything out about it, I'm going to ask you to just fill out a very short report and then paste your final sequences here. So you put your name, whichever transcriptome you are working on, just so I know and I can compare. Each transcriptome will have 3 or 4 students working on it. So I was transcriptome 0. And then answer these questions. If you need help, if you'd like to log on to an office hour or set up an appointment with me, I can do that. And then lastly, just cut and paste. If you've got a mega file, you can actually just highlight, do Control A or highlight all of them like this. And then you can just use the Control C and then Control V, and you can paste your sequences. It's going to make it a little long, so we may have a couple extra pages, but that's fine. And for each one, we'll have a name and then a sequence following the name. Okay, so do that, and I'll have the sequences. And then once you get done, save that, and then just attach this to the assignment. And I'll create one little assignment. All you have to do is log on, attach the file, and then you're done with project number 4. Now, I am going to do one other thing. You don't need to do this, but just to kind of demonstrate and show you what can be done and what we may want to do with some of these. And I'll only do it with one of them. Let's take the insulin receptor. So I'm going to copy just the insulin receptor here. And then what I'm going to do is I'm going to go back to my original folder or file with these genes in it. So let's get rid of the other 2 receptors. And now I'm going to paste in my found insulin receptor, potential insulin receptor. And what I want to do is compare it to my original and see how similar they really are. Now notice it's out of reading frame. I might have a whole transcript without just the coding region. So what I'd like to do first is translate these into the protein sequence. And notice that I have lots of stop codons in that top sequence, that there's really not any long sequences without stop codons. So it's very clear that I'm not in the correct reading frame. So I'm going to just add a space to shift the reading frame and then look again. And I'm going to do that for all 6 reading frames to see if I can find the correct reading frame. Now, there are programs that will do this that are automated, especially if you have lots of them to do. You can look into getting those, and it will just output the longest open reading frame, and then you could use that, and we wouldn't have to go through this. But notice that when I shifted over just a single base pair, that suddenly I don't have any stop codons in this top reading frame. So that's very, very clear. Oh, great, I found it. Still doesn't look very similar, most likely because it just hasn't been aligned. It may be divergent anyway. But we have 2 insulin receptors here now in their proper reading frame. So now I can say, let's align these at the protein level and see how similar they look. So here we go. And for some reason, let me try one more time. It doesn't look like it did it. Let's select. Oh, I think what I had, it just had one residue highlight. Let's highlight all of it and do that alignment again. There we go. That's a little better. So notice that there's definitely a difference, right? The locust one is much larger, but where they do line up, and that may very well mean, so it doesn't look like you even have a stop codon. So very likely I just have a partial of this gene, and I don't have the full length sequence. But notice that where it does line up at the protein level, it is remarkably conserved. Now, at the DNA level, we could come and look at it and say, well, let's see if what's going on there. And still we have much more diversity, right? So there is a lot more. And so we may have some pattern of both non-synonymous and synonymous mutations going on here. But let's just kind of go in on a little area. So notice that when I unhighlight it, there are lots of changes there. But when I look at that at the protein level, it's much more conserved. We still do have differences in proteins, but you can see that in many areas, those amino acids are the same. So we probably have a lot of 3rd position mutations. We probably have some 1st and 2nd position mutations, and we could evaluate those, do Ka to Ks ratio, and look at some of those aspects. And what I will do is once everyone's turned these in, I will probably go ahead and put all of the 8 different receptors into an alignment like this and see if we can see any patterns across the entire group, see if we can see anything that's interesting there, and then I may post a very short update. So that's it for project number 4. Make sure you complete everything. The only thing that you need to submit is this final project 4 report complete, and then attached to the assignment that you will find there in the same folder, along with the link to where all of these files were.